So I want to cover sperm production. And to first do that, we're going to actually have to talk about the testicle and the anatomy within the testicle. So you've got your testicle, and sitting on top of that testicle is a structure that is eventually going to do sperm storage. And this is called the epididymis. All right, if we look at within the testicle itself, it's got kind of segments, and those segments are not, I don't know, they look kind of like an orange, I guess. And within those, there are these long squiggly tubes. Those tubes are called seminiferous tubules. And in those tubes, that's where the sperm production is actually going to take place. If we were to actually dissect one of those seminiferous tubules, so I'm going to grab a little piece of one and bring it down here and make it really big. What you'd see inside that tube is you'd see at the base sort of germ cells. Germ cells are like stem cells that are going to be making the sperm. And then next to those stem cells that make the sperm, you've got, they're called sustentacular cells, and they're the ones that are going to be supporting the sperm. They're giving it sustenance, so that's where that comes from. So sustentacular and germ cells, which are like sperm stem cells. Okay, so these sperm stem cells are going to be constantly making sperm, and as they're making sperm, and it's going to go through the whole process to become sperm, the cells that it's making are going to be going further and further towards the center of this tube, and then eventually they're going to be sort of pushed out of the tube because, well, they'll be able to swim. So how does that process happen? Well, the first thing that happens is that you've got, I need a new piece of paper. So what do I expect you to know about how do you make sperm? Well, the sperm stem cells, or germ cells, are called spermatogonia. And these guys are going to make sperm throughout the majority of a man's life. The first thing that happens is these spermatogonia undergo a mitotic division. So you get two identical cells. One of these will continue developing, and that one we call the primary spermatocyte. And the other is going to basically hang out and stay as a spermatogonium clone. Okay, that primary spermatocyte is going to divide again. This division is a meiotic division. So now it will only have half the DNA it did originally. And this is where it's a secondary spermatocyte. And it's making two of those. Now, meiosis wouldn't be complete without doing two more divisions. And these divisions are going to create spermatids. And what I want you to notice is that we've gone from one spermatogonium to 1, 2, 3, 4 spermatids. And what's going to happen to those spermatids is that they're going to continue differentiating into sperm. Basically, they're going to go from being a cell to becoming a little sperm. And as they're developing into the sperm, they're going to get this, we call it a flagellum, that helps them to swim. They'll have this little area by their tail, which is full of mitochondria. And then they're going to have the head, which is genetic material. And also this little area at the tip, which is called the acrosome. The acrosome is actually filled with enzymes, and those enzymes are going to help the sperm to get into the egg because they're going to digest the zona pellucida, which is the outside layer of the egg, and allow the sperm to inject its genetic material into that egg. So I can anticipate a question about the hormones and what's going on with the hormones. Well, remember that you've got gonadotropin releasing hormone, and this is going to be coming from the hypothalamus in the brain. And that's going to work on the anterior pituitary, causing the release of follicle stimulating hormone and luteinizing hormone. Now, okay, those are going to work on the testicle. And when those reach the testicle, what's going to happen is the luteinizing hormone is going to cause the some of the cells that are within the testicle, the interstitial cells, but you don't need to know their exact name, to secrete testosterone. And that testosterone is actually going to stimulate sperm production. Meanwhile, what's going to happen is that testosterone is also going to inhibit that gonadotropin releasing hormone. And the reason it's doing that is that it's kind of saying, hey, I'm in sperm production mode. I don't need to stay in, I don't need to make infinite sperm. I guess you don't want the testicles to get too full. Meanwhile, what is the follicle stimulating hormone doing? Well, this guy results in the secretion of another hormone that's called androgen binding protein. And androgen binding protein, it kind of keeps the testosterone in the testicle. It puts the test in testicle. Sorry, it's late and I'm being goofy. Okay. So now as the sperm levels continue to rise, they're going to cause the release of a hormone called inhibin, which sounds like what it does. It's an inhibitory hormone. And inhibin actually is going to inhibit follicle stimulating hormone. By inhibiting that, what happens is now the testosterone is not local anymore, so it's kind of dissipating, and since it's dissipating, now we're not going to have as much sperm production. So these are ways that the inhibin and the testosterone's inhibiting GnRH are both things that are going to decrease the amount of sperm production, albeit in different ways, right? GnRH is going to sort of decreases testosterone production, and inhibin is going to inhibit the holding of the testosterone in the testicle, which means it can kind of go wherever. And if it's going wherever, then there's not going to be as much working on those stem cells telling them, hey, you need to make more sperm.